Welcome to the Lipid Maps podcast. This episode has been sponsored by Avanti Research, part of Croda Pharma. We're grateful for their ongoing support of Lipid Maps. Today we're joined by John Harwood, who is Emeritus Professor here at Cardiff University. John, it's very nice to talk to you today.
Okay, thanks, Matt.
You started your research career in medical work in the 1960s, but then moved into plant lipid metabolism alongside more general animal lipid metabolism too. How did you do research into lipid metabolism back then? It must have been very different to how it's done now.
Yes, indeed. Perhaps I could just add a little historical note. For my PhD, I was working at Birmingham University. I was working on inositol lipids, which are quite famous now. But when I was working on them, they were just newly discovered, really, and a very exciting area to work on. And I then wanted to do a postdoctoral in the United States, which was the custom in those days. I mean, the United States had a much more vigorous research culture at the time because Europe was recovering from the Second World War. So it was, as I said, it was the main thing to go to the United States. And I wanted to go to the West Coast. University of California was a very good place, a very good university in general. And I was offered two positions, one in Los Angeles and one in Davis, California. And the one in Davis, California was to work on plant lipids, which I'd never worked on before. And not only that, I'd never even had a lecture on plant biochemistry, not even on photosynthesis. So I was completely a novice in this area. But Paul Stumpf, who was the person I wanted to work with, was a well-known figure. And furthermore, somebody I knew had already worked with him and said he was a great guy to work with. So I went off to Davis, California to learn about plants and started my research on plant biochemistry. Now you ask how things have changed over the years. And I had to think about this. I mean, lots of things have changed over the years, of course. There are lots of advances. Probably the main things that occurred to me was, first of all, there was a breakthrough just before I started my PhD, and that was the discovery of radioisotopes and how you could use radioisotopes to study metabolism. And in particular, P32 was an isotope which was available. And if you were working on phospholipids, then P32 was a very good isotope to use. I should say it was quite dangerous. It's what is called a hard beta emitter, so it forms beta rays which are quite penetrating. We had to use lead bricks to sort of shield all our solutions of P32, and you had to be very careful that you screen them away to stop radio damage. Radio damage obviously was very prominent with somebody like Marie Curie, who discovered radioisotopes. Of course, she suffered from the problems with radio damage. So that was the first thing. Radioisotopes could now be used to study metabolism. And it meant you could study reactions which were not very active. Very small reactions could be studied with radioisotopes. The other thing that changed over the years, or two other things that changed over the years, one was the method of separating lipids. For my PhD, for example, I used a lot of paper chromatography. Yeah. You would separate the lipids on paper. You would cut out the spots which were radioactive. You then had to digest them with perchloric acid, which in itself was dangerous. You could get explosions if you weren't very, very careful.
How often did you have explosions?
Well, in the department, we probably had one explosion a year.
Right.
And it
not infrequently.
Yeah. Well, I mean, you would carry out the reactions in a fume cupboard, and it would blow the fume cupboard up. Mm-hmm. So you would have to replace the fume cupboard, and so it really was quite. It wouldn't be allowed nowadays, I'm sure.
No.
But anyway, you had to get a clear solution in order to use a Geiger counter to count the radioactivity. So we had paper chromatography. We did a little bit of thin layer chromatography, but modern methods such as liquid chromatography coupled to mass spectrometry for example, those were big advances, and they were another 20 years before they came along. The third aspect, which has changed completely over the years, is molecular biology. The double helix had only just been proposed when I was a student at Birmingham University, Watson and Crick and so on. So, and then, of course, people discovered that there were codes that coded for the various amino acids, and those were deduced in the next 10 years or so when I was a student and then a young postdoc. And then, of course, actually looking at sequences in genomes and so on, that's been revolutionized really over the last 20 years. It was a very difficult thing to do originally. It was very expensive. And now we have modern methods which in some countries where they have a bit more money than the UK, they routinely get complete genome sequences for individuals. So those are the three areas. And I should say also the improvements in chromatography, which have come about using GC-MS and using liquid chromatography MS, they're much more sensitive than the old methods that we used to use. You can really detect changes in different compounds down to a very, very low level, micromolar levels and so on.
So minutely sensitive now you can...
That's right. And of course...
I saw a paper recently that reminded people that they may not be measuring their sample. They may be measuring plasticizers that had been diluted from the
That's right.
glassware or the plasticware.
That's right. I mean, even with gas chromatography, gas chromatography had only just really been developed when I was doing my first postdocs. And one of the advantages of going to Stump's lab is he had the GCs, gas chromatograms, that, were very sensitive at detecting individual fatty acids, for example, but he had them linked to a radiochemical detector, and the carbon was oxidized to carbon dioxide, and that was then counted in a counter that was attached to the GC. So it was radio GC that we were using, and that was very, very useful because you could discover how fatty acids were being made.
And now, of course, we do that by mass spec rather than radio counting. Radioisotopes are used far, far less now than they were in those early days.
Indeed. When I was doing my PhD, we were working on benches, and the benches had two sides with sort of reagent bottles in between. And so on the opposite side of the bench I was working on, there was a sabbatical professor who was visiting the lab. And I was monitoring my bench to make sure I hadn't spilt anything with a Geiger counter, and the Geiger counter went off scale. And I couldn't understand what had happened because I was usually quite careful and I hadn't spilt anything. And what had happened was that the sabbatical professor had a flask of radioactive ATP, and he had very carefully screened around the flask with lead bricks to protect himself, but he hadn't screened my side of the flask.
You were being irradiated.
So I was getting all the radiation. Yes. Yeah. And it turns out actually that lead bricks weren't really very good because they produced alpha rays, which are even more hazardous. So, things that you discover as you go along.
So would you say you've had any defining theme in your research career?
I've always been interested in the unknown. Discovering new lipids, characterizing them, and then asking the question, how are they made? Some of my early work in Stump's lab, and later on, we discovered new enzymes that weren't known for fatty acid biosynthesis, for example. Several new enzymes were discovered. And then having discovered how individual lipids were made, you then ask the question, what are they doing? What is the function of these lipids? And so that's the overall theme. Whenever I've found a lipid, I've asked how it's made, how it's biosynthesized, and how it's broken down. And then what's it doing? What's the function of these lipids in different organisms? They may have different functions in plants that they have in mammals, for example. Yes. So, those are the overall themes. And so that's led me to work with lots of different organisms and also work with different people. And I should say, I'm a great believer in collaboration. I think if you find a particular lipid which you think might be involved, for example, in cartilage degradation and arthritis, the best thing is to find somebody who is an expert on arthritis and collaborate with them. You can bring your lipid expertise and they bring other expertise, and the sum of the two is always better than just one person. So I've always been a great believer in collaboration, and I've been fortunate that by and large, I've met people who have been very willing to collaborate. When I first started out my scientific career, a lot of people were wondering how ATP was made, but they knew it was made in mitochondria, for example. And the so-called mitochondriacs were people that specialized in mitochondrial production of ATP, and they were terribly vicious to each other, always arguing with each other, asking awkward questions in conferences and so on. Fortunately, in the lipid area, by and large, there have been one or two difficult moments, but by and large, people have been very friendly and cooperative, and so I've been lucky to be in lipid metabolism.
And as well as plants and the animal work, you were telling me that you did quite a lot of work on pulmonary surfactant lipids. Tell me about that. When I first got a position as a lecturer in Cardiff, I was looking for some relevant areas within South Wales. And South Wales, of course, used to have a lot of coal mines, a lot of quarries and so on. And so lung diseases were a real problem. So I started to look into this, again, with people in the MRC units at the local hospital. We were interested in pulmonary surfactant. Now, this is a lipid where it's a lipoprotein mixture, about 90% lipid, 10% protein, and it is secreted into the lung. And if you think about it, when you breathe, you take in air and then you breathe out. And when you breathe out, your lungs remain partly inflated. It's not like a balloon where you let the air out of a balloon, it just collapses flat. If that happened in the lung, you would have great difficulty getting your next breath, because you'd have water-lined tissue pressed against each other. And if you've ever tried to separate two pieces of glass with water in between, it's very, very difficult. Be super glued together. Now, the saving grace is that you produce this lipoprotein mixture, pulmonary surfactant. Produce it as a child, as an infant, in the last month of gestation period. In the last month, the baby produces its own surfactant. Up until then, it doesn't. So if it's born prematurely, the danger is it hasn't got pulmonary surfactant and it can't breathe properly. And so this was an area where, there was a lot of deaths. We suggested you could produce artificial surfactant, and we produced some artificial surfactant. That could be then intubated into the baby and help it to breathe for 1 or 2 days until it produced its own surfactant. And this was very successful, actually. Various artificial surfactants are now used routinely for prematurely infants in the UK. So it's saved a lot of lives. And that was very gratifying to work with newborn babies, have a method for keeping them alive. And you said that the work on that was triggered by the mines, all the dust in the mines and the miners.
Well, we were looking, that's right. We were looking at lung function in general, and then we isolated lung surfactant and so on. Well, I often find that, something triggers your interest in things, and that then leads to further work. I mean, for example, I've done quite a lot of work with arthritis. From personal reasons. I've suffered from arthritis myself. I did too much running when I was young. And because of that, I worked with people who worked on cartilage metabolism. And we discovered in particular that certain fatty acids were very useful. And we actually discovered it from some work with the US Army. They had dogs, beagles, that were suffering from arthritis. And we suggested that you could feed them this particular type of fatty acid, and that might be a benefit. And it was within a month, these beagles recovered from limping to being fully fit. The actual fatty acid that was beneficial were the so-called omega-3 polyunsaturated fatty acids. They're present in fish oils, for example, and so on. And you'll find if you go around the local supermarket, there are lots of oils which contain omega-3 fatty acids, and this is considered to be beneficial for people. And it's also added routinely to dog food as well. Again, an accidental observation, a personal observation, did a bit of work on that. And in fact, omega-3 polyunsaturated fatty acids are considered very important for a number of diseases, heart disease, dementia, and so on. So there's a lot of interest in this particular area. And of course, because I work on plant fatty acids, I'm interested in how plants make them. We can't make omega-3 fatty acids ourselves. We have to rely on ingestion of these in the diet. Usually, as I said, fish oils, and fish don't make them either. Fish eat algae, which do make them. Yeah. And so you've got a sort of match between the plant and algal side of things, producing these polyunsaturated fatty acids, which are then used for health reasons.
In 2016, you were awarded the Biochemical Society Morton Lecture, reflecting outstanding contributions to lipid biochemistry. And they said there how much the demand for lipids has increased over the past 50 years, not just for diet, but for industrial processes as well. Your more recent work has involved modifying plants to change their lipid composition. So is that to increase the amount of lipids for industrial use, or to change the amount that are beneficial for health? Could you tell us something about that work?
Yeah, well, edible oils are very important in the diet. So we need to have a balance of protein, carbohydrate, and fat. And of course, as I've just said, we need to have certain fats because they're required for good health and we can't make them. With increases in population and increase with beneficial diets, the demand for edible oils has gone up about 5% a year for the last 50 years. So there's a continuous increase in the demand for edible oils. There are 4 crops which between them actually provide about 90 to 95% of the total oils. And of these, palm oil produced by the oil palm tree represents, well, about 45% of the total oils that we consume. We could not possibly stop using palm oil because it's so important. So that is not a scenario that's realistic. The other thing is we have to be careful about clearing primary forest to make way for palm plantations. That is being carefully monitored. And because the total amount of agricultural land in the world is limited, the only way that you can really increase the amount of edible oils that are being produced is to make the production of them more efficient per acre or per hectare. And the other advantage with palm oil is that it produces about between 8 and 10 times as much oil per acre as any other oil crop, because most oil crops are annual. You sow them in the spring and then you have one harvest, whereas the oil palm is producing oil all the time. So it's much more efficient. So that's another advantage there. So we've been looking at the possibility of increasing the total amount of oil that can be produced by a given crop. We've worked on oil palm, we've worked with soybean, we've worked with oilseed rape, which is relevant to Britain. So we're trying to increase the amount of oil that's produced, the efficiency of production. There are ways, we haven't concentrated so much, but there are ways in which you can modify the nature of the oil that's been produced by genetic modification. That's not very popular in Europe at the moment, but nevertheless, I think it will be in the future. But we've in particular looked at increasing the total amount of oil being produced, and we've used a new method, which is called flux control analysis. So this measures the flux, that is the movement of carbon, down the various biosynthetic pathways to produce oil. and you can measure it quantitatively using flux control analysis. So we pioneered that particular type of technique, well, on lipid metabolism in general. We're the first people to do it in plants for biosynthesis of lipids. So it's a useful quantitative method that you can measure. If you do a manipulation, what effect does it have on the total amount of oil being produced?
So that's been really interesting, John. We've had a really good look through from exploding fume cupboards and radioactive shielding through to arthritic dogs and saving babies' lives to improving the efficiency of oil palms. It's been a really interesting look back at your career.
Perhaps I could just add something. You mentioned the Morton Lecture Award that I got. But I've had a number of awards from the Americans, the Supelco Medal, and in particular the Stephen Chang Award. And the Stephen Chang Award is given to people who've used basic biochemistry and chemistry to actually produce products which are then useful in industry. So it's the application of basic research to industry. And I was awarded this partly for work on surfactant that you've already mentioned, but we work with herbicides a lot. And we've worked, of course, as I've said, with flux control analysis. So that sort of is an application of somebody in a university doing basic research and actually applying it to what is sometimes referred to as a real world situation, i.e. what industry and what the food companies are actually doing.
And that's a good reminder that there is always value in basic research because you never know where it might lead to.
Oh, absolutely. Absolutely.
So thanks a lot, John, for taking the time to talk to us today. And thanks to all our listeners. And we hope you'll join us again for the next Lipid Maps podcast sometime soon.